Hello, I'm Ayse Dincer. I'm doing my PhD in computer science at University of Washington. Today, I want to talk about my project, Adversarial Deconfounding Autoencoder, for learning robust gene expression embeddings. Gene expression profiles provide a snapshot of cellular activity, which makes it easier for us to understand the associations between disease, expression, and environmental factors. And recently, more and more expression datasets are becoming available, making it possible for us to examine complex nonlinear models, such as neural networks, on various biological problems. And among these models, I believe that unsupervised deep learning has enormous potential to extract important signals from gene expression profiles. By the way, as a super quick reminder, unsupervised neural networks, or autoencoders, consist of an encoder and decoder network with a bottleneck layer in the middle. And this model is trying to have an encoder that takes the input features to generate an embedding such that we can reconstruct the input features as successfully as possible. And these unsupervised models are promising, I think, because first of all, they give us the ability to train informative models without supervision. This is really useful because you would all know how difficult it is to come up with a high number of samples with coherent labels. And the second reason is because with these models, we can extract patterns without any imposed directions or restrictions, which can actually be the key to learning something new about the biology. So it sounds super promising, but unfortunately, it is not so straightforward to employ these models on gene expression data because expression measurements often contain other sources of variation in addition to the true signals we seek, especially if you have samples from a large cohort or multiple cohorts. Then you can see variation as a result of out-of-interest biological variables such as age or medications used, and technical artifacts that are not related to biology, such as batch effects. In our project, we call these biological or non-biological artifacts that are systematically affecting our expression values as confounders. And the problem is that in many datasets, these confounder-based variations might mask true signals and make it difficult to learn reasonable patterns. An example of this problem, here we have a breast cancer expression dataset from two different studies. And in order to examine the strongest source of variation, we plotted the top principal components of the expression matrix. And naturally, we observed that the data set is a very strong effect in this space. And then we simply passed this expression matrix to an autoencoder and generated the same plot for the embedding. And again, the principal component plots showed us that this dataset effect present in the original expression space simply translates to the embedding space, which can be quite problematic. But even more important problem is that confounders can prevent our learning robust transferable models. By a transferable model, we mean that if you learn a signal from an expression dataset, such as a disease signal, for instance, you would want this signal to be valid for reasonably similar datasets. So to simulate this problem, we simply trained our autoencoder on two datasets and then transferred to an external dataset unseen by the model. And this principal component plot clearly shows you that the external samples are separated from the training samples, showing us how when you have confounders, it will be difficult for you to learn transferable models. So in our paper, we want to address this problem of entanglement of confounders and true biological signals. And with this, we want to generate biologically informative expression embeddings that are robust to confounders and are generalizable so that we can hopefully show the power of deep unsupervised models. And to achieve this, we propose our deep learning approach, which we name as Adversarial Deconfounding Autoencoder, ADAE in short. ADAE is actually an extension of an autoencoder. So just to remind everyone, an autoencoder contains an encoder network which maps from input expression space to an embedding Z, and then a decoder that maps the embedding Z back to the input space. And we optimize our encoder and decoder networks in order to minimize the reconstruction error. ADAE consists of two networks. The first one is an autoencoder model that is optimized to generate an embedding that can reconstruct the original input. And the second model is an adversary model that takes the embedding generated by the autoencoder and tries to predict the confounder. And our goal is to learn an embedding C that can encode as much information as possible while not encoding any confounding signal. And to achieve this, we train these two models simultaneously. The autoencoder tries to reconstruct the data, but also tries to prevent the adversary from accurately predicting the confounder. But at the same time, the adversarial predictor is still trying to update its weights to obtain a better performance. So more specifically, we train our model in 3 steps. The first step is just pre-training the autoencoder to generate an informative embedding C. The second step, naturally, is to pre-train the adversary. So here we define a general loss function L, which can be selected based on the confounder you have. Like for instance, it can be cross entropy for a categorical confounder. And lastly, but most importantly, we begin joint adversarial training by optimizing over the 2 networks. So to do that, we first freeze the weights of the adversary, then train the autoencoder model for 1 epoch. So this means that we're trying to minimize the autoencoder loss while maximizing adversarial loss. We then freeze the autoencoder and train the adversary model for another epoch to this time minimize the adversarial loss. And we basically continue this alternating training process until both models are optimized. Oh, by the way, as you can guess, this lambda variable here determines the ratio of weight the model gives to reconstruction or deconfounding. So increasing this value would mean learning a more deconfounded embedding while sacrificing reconstruction loss. So we aim to demonstrate the broad applicability of our model by employing it on different expression data sets and experimenting with different cases of confounders. And to evaluate, we checked, first of all, the deconfounding of the learned latent space, then preservation of the biological signals, and then lastly, we check how the prediction performance changes when we transfer our embedding from one confounder domain to another. So for this presentation, I'm just going to focus on our experiments on K-unplot breast cancer expression, which combines multiple studies, and we use the dataset label as the confounder variable. But we have more experiments that I would love to talk about later. So to show that ADAE can learn biologically meaningful deconfounded embeddings, we generated UMAP plots of embeddings with a standard autoencoder and ADAE, and simply colored by the dataset label. So we first of all observed that for a standard autoencoder embedding, naturally the datasets are clearly separated from each other, showing us that the space is highly confounded. But on the other hand, ADAE embedding can clearly fuse the data points from different datasets, and therefore eliminating this obvious dataset effect. But more interestingly, we tried coloring this UMAP plot by different biological variables of interest. So we tried estrogen receptor status and cancer grades, which are important phenotypes for determining cancer treatment. For the autoencoder embedding, we can see that the samples are not very clearly differentiated by the phenotype labels, while ADAE can clearly manage to separate these samples by both phenotypes, again showing us how confounder signals might sometimes overshadow biological signals, and once removed, it can help us learn more meaningful patterns. So here, another feature of ADAE is that it can generate embeddings that can generalize to different domains. And the most direct example of this would be to learn an embedding from a dataset and just transferring it to a separate dataset. And we simulate this with our breast cancer expression samples. We just simply left one dataset out and trained the autoencoder on the remaining datasets. And then with these circle markers, we're showing the embeddings learned for the training samples. And with the diamonds, we show the embedding for the external samples. So first, for the standard autoencoder embedding, we notice that the external samples are clustered entirely separately from the training samples. On the other hand, when we examine the same plot for ADAE, we observe that the training samples and the external samples are highly overlapping with each other. It's almost not possible to distinguish them. But more importantly, you can see that the direction of encoding of cancer grade for the training samples is also valid for the external samples for the ADAE embedding, which is totally not possible for a standard autoencoder. And I think this is a very interesting example showing us once you deconfound a latent space, then you can learn more transferable manifolds of the data. We also wanted to quantitatively evaluate our model by comparing it to alternative deconfounding approaches. And to do that, we fit prediction models on the ADAE embeddings and other competitor embeddings to predict various biological phenotypes of interest. And we evaluated the performance of the models on an internal test set as well as an external test set that they were not trained on. Here we show the great prediction performance of ADAE and alternative deconfounding approaches. So we compared ADAE to not corrected standard autoencoder model as well as some alternative deconfounding approaches. And we observed that for the external test set, ADAE can outperform all other models. But the more interesting result, in my opinion, is that we calculated the generalization gap, which is the distance between the internal and external test set performances. So a high generalization gap means that the model performance declines sharply when transferred from an internal test set to an external test set. And we observed that this generalization gap for cancer grade prediction is much smaller for ADAE, which basically tells us that when you transfer your model to an unseen dataset, you have a minimal performance decline, which is actually what we want from a robust embedding. We have many other experiments on ADAE and on different data sets and different confounder settings. For instance, we experimented with sex and age, and we also carried really interesting transfer experiments where we transfer across different confounder domains, such as we trained on male samples and then predicted for female samples. And we also transferred across different age distributions to basically test the limits of transferability of ADAE. So to conclude, we introduced Adversarial Deconfounding Autoencoder to generate expression embeddings that are robust to confounders. And with our experiments, we tried to show that ADAE can learn deconfounded embeddings that can preserve biological signals and can transfer across different confounder domains. In the future, we want to experiment with regularized autoencoders, such as variation autoencoder, for instance, to further regularize the latent space. Another interesting idea is that we want to correct for multiple confounders at the same time, for instance, correcting for age and sex at the same time. And most importantly, we want to explore the broad applicability of our model by applying it on new data sets, adjusting for new confounders, and hopefully employing this computational tool on new diseases to understand the underlying biology. So we're very excited about this project, and I hope you became excited as well. Thank you very much for listening. I would love to talk further.